All right. Again, what I wanted to cover in this figure here is where carbons come in and where they go out. So here we have acetyl-CoA coming in, and we're going to mark it in blue. Uh, then we have this combining with oxaloacetate, which has two carboxylic acid groups. We're going to mark that in green, okay? So when this carbon comes in, we are hydrolyzing, which is allowing us to gain another oxygen and a proton, and we are essentially popping this on in a slightly backwards method. So we're attaching in a different location, which leaves us with a carboxylic acid group originating from this acetyl-CoA. As we get down here where we are losing our first CO2, recognize that we are actually losing the CO2 from this middle carbon, which originated from oxaloacetate, okay, leaving this compound here, alpha-ketoglutarate. Then again, when we go through the next round where we lose the second CO2, recognize that the CO2 we're losing is the other carboxylic acid group from oxaloacetate, not the one originating from acetyl-CoA. That carboxylic acid group and the other carbon associated with it is still present, okay? And when we progress from succinyl-CoA to succinate, we lose track of which carbon is which because succinate is a symmetrical molecule, right? So there is no one end or the other that we can tell which end is which. Consequently, when this enzyme or when this compound goes into the next enzyme, it could go in one of two different orientations. Consequently, as we progress through and recognize that one of these carboxylic acid groups present ultimately on oxaloacetate originated from the next cycle back of acetyl-CoA, so it's present here, 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 here. It's one of these two, it's one of these two, it's one of these two, and it is now one of these two, okay? So now, when we go back through, it takes at least 2 turns to get rid of one of these acetyl-CoAs, or one of these CO2s originating from acetyl-CoA. Or it could take 2 more turns, right? Since this is symmetrical and we can't tell which one is which, right? So this can take multiple turns in order to ultimately liberate these carbons as CO2. Now, just like glycolysis, we have particular steps which are high energy steps, and those steps tend to be where key regulatory elements come in. So going from pyruvate to acetyl-CoA is an energy expensive proposition. Consequently, if you have a lot of ATP already, you don't need to do this. If you have a lot of NADH, you don't need to do this. If you have a lot of acetyl-CoA, you do not need to do this, right? So these are all things that inhibit this enzyme. On the reverse side, if you have a lot of pyruvate, or you are being signaled by insulin, or you have more ADP than you have ATP, this is going to stimulate this reaction, okay? So all along here where we have these key high energy steps, we have opportunities to introduce regulation. A second thing to consider with the citric acid cycle, it is not only about producing energy, right? So mostly what we think about is citric acid cycle producing electrons in the form of NADH to move on to the next step. This isn't the only thing it does. There are a number of compounds that originate out of this cycle. So citrate is a key starting compound for fatty acids and sterols. Alpha-ketoglutamate is the addition of one amino group to becoming glutamate, right? And this can be turned into other amino acids. Over here, oxaloacetate is one step away from being aspartate, which is a key source of other amino acids and is also a source of some of our nucleotides. Um, and so you can see that there are different things that come out here. So succinyl-CoA is actually a starting point for the heme group in our hemoglobin. It is also the key starting place for a very similar compound that is in chlorophyll, or a very similar ring-like structure um, that we have in hemoglobin. So there's a lot of other things that this process gleans. So it's not only about energy production, it's about key starting compounds to make other things as well. So consequently, you may not need to do this cycle for energy, but you may need to do this cycle to make more hemoglobin, or you may need to do this cycle because you need more aspartate or glutamate or all the various amino acids that come out of here. So we have summarized the reactions of the citric acid cycle. Again, I will cover or have you do the citric acid cycle in a quiz like we did in glycolysis, and I will not have you do this on the exam. However, since I have talked about some of these key compounds, I would want you to recognize where they're falling in. So that is kind of fair game for the exam.